Hello to our friends joining us via recording. Today we are talking about much of the content from lesson number 7, which is about the skin. Remember that the fancy word for the skin is the integumentary system. So we are diving into the integument, or we're diving into the skin this week. First thing we're going to do when we look at the layers of the skin is we're going to remember for our lab exam what kinds of tissues are found in each of the places. So I'm going to start with this tissue over here on the left. When I look at this tissue, notice that I see these big open cells with little black spots, well, purple spots really, on the sides of these cells. When I'm talking about this kind of cell, can we identify the type of tissue that we're seeing here with these large cells? They look empty, but they actually wouldn't be. It's mean for me to make you type this morning, isn't it? Yeah, so we've got a couple of options that have been listed here in the chat. The correct option for this one is the adipose tissue, but I'm really glad, Seth, that you mentioned the other one that this looks like, because it's really important for our exam that we're able to compare those two. So what we're looking at right here on this slide is adipose loose connective tissue. So this is adipose tissue. Adipose tissue is found in the part of the skin called the hypodermis. Some of the other places you learn for lab that we found adipose tissue is around organs. This is the kind of tissue that prevents heat loss. It cushions our organs. Adipose loose connective tissue, part of the skin. What makes this different from simple squamous is notice that when I look at a cell like this one here, I've got a lot of places where all I see is kind of this light pink. This would be the plasma membrane of the cell. And it's the same deal here, the plasma membrane of the cell. If this was simple squamous epithelium, which I'm guaranteeing you will see on the lab exam, I'm fairly confident we'll see it, what would be different, let me get my pen here, is we would see more nuclei. So if I had nuclei all over the place, mixed in, across these membranes, if I can't hardly go any space without another nucleus, that would be simple squamous. It looks very similar to this though, right? Because when we talked about simple squamous epithelium, that was found in the lungs. So in the lungs, these spaces would be empty. That'd be where the air is. Since we're in adipose tissue, since we're talking about a type of cells called adipocytes, I just put that in the chat for us, adipocytes, this in living tissue would be filled with lipids, because this is a fat storage tissue. So the first tissue we see on the left is in the hypodermis layer of the skin. Next door neighbors to the tissue on my slide here, this slide tissue that we're looking here, this is actually on the outermost layer of the skin. Can you help me out in the chat? When we're talking about the epidermis, you totally don't have to type the whole name because it's really long, right? Let's start with the main tissue type. Is this epithelial, connective, muscle, or nervous? Let's start there. When I look at this type of tissue, which of those 4 main types are we seeing there? Epithelial, connective, muscle, or nervous? What do we think? Yeah, several of us are chiming in. This is an example of epithelial tissue. The way we can tell that is when you look at this tissue, all of this is cells. When we talk about epithelial tissue, all they have is cells. There's no proteins on the outside, and the cells are very small. So each of these purple dots that I see down here represents an individual cell. And remember, in this tissue on the outside of the skin, all of these are cells as well, cells that are filled with keratin and are dead. So all kinds of cells all over the place. We're epithelial tissue. Yeah, so Hugh took one for the team and he typed our whole big long name. So this is epithelial tissue, which means we name it based on the number of layers that we have and based on the shape of our cells. So when I look at this tissue, it should hopefully be pretty obvious to us that we have more than one layer of cells, lots and lots of layers of cells in this tissue. And when I look at those cells, remember that we have to look at the apical surface, the top surface. We see that these cells are really flat. So more than one layer of cells is called stratified. When we talk about the squished cells, those are called squamous cells. The last part of its name tells us that all these cells on the outside are filled with keratin. So this is keratinized, stratified, squamous epithelium. I'm not sure if I emphasized it very much in our lab classes, so I want to make sure to take a moment for this question here. When we talk about the kind of cells that are found in keratinized, stratified, squamous epithelium, this is the kind of cells that get their name because they're filled with the keratin protein. Does anyone happen to remember what I call these cells that I see in here? Exactly. Yeah, several of us chiming in. The name of these cells is keratinocytes. Keratinocytes, meaning I'm a cell that's filled with keratin. Remember, so we talked about adipocytes over here on the left. When we talk about this kind of epithelium, we have the keratinocytes. And when we flip over to my connective tissue over here, I have a special kind of cell that gets its name because it builds protein fibers. Does anyone remember the name of these cells, these purple spots that I'm seeing here in my tissue here? Yes, exactly, fibroblasts. So fibroblasts are found in pretty much every type of connective tissue that has protein fibers. We can see them best in this kind of connective tissue that I'm looking at right here. This is areolar loose connective tissue. So when I look at areolar loose connective tissue, the kind of cells that I see inside of it are the kind of cells that can build protein fibers. So these guys are called fibroblasts. They're fiber builders. When we're talking about the keratinized, stratified, squamous epithelium, these are keratinocytes because they're full of keratin, or adipocytes because they're full of lipids. They're in adipose tissue. When we talk about the areolar loose connective tissue that I see here, this is the kind of tissue that I find in the place of the skin that has those up and down bumps. What was that upper part of the dermis called with those up and down bumps? Or in other words, what's the specific location of areolar loose connective tissue? Where do I find that one? Yeah, exactly. The up and down bumps in the skin, these are called the papillary layer of the dermis. So areolar loose connective tissue is what I find in the very top of the skin, going up and down, where the connective tissue meets the epithelial tissue on the outside. One other kind of tissue that we have in the skin, this is the kind of tissue that's found in what's called the reticular layer of the skin. This kind of tissue here on the right, can you tell me in the chat, what kind of tissue are we looking at here? This really messy pink one that has these pink proteins all over the place. Exactly. Yep, so this is an example of dense, irregular connective tissue. Dense because everywhere that I look on this slide is pretty much covered with proteins. Irregular because some of these proteins are sticking out of the screen at you. Some of these proteins are going sideways. Some of them are in at an angle. It's a hot mess, so it's dense, irregular connective tissue. Remember that we also have in the body dense, regular connective tissue. And when you look at that one, all of the proteins are arranged the same way. So if you're looking at a tissue where all the proteins are going the same direction and all I see is lines and lines of proteins, dense, regular connective tissue. How do we feel about the tissue types of the skin? Give me a thumbs up or any questions that you have for me in the chat. Yeah, Amush is totally right. This kind of tissue, we do find it in the reticular layer of the dermis. That's absolutely right. Yep. Next door neighbors, areolar, we find that in the papillary layer of the dermis. Perfect. Lots of thumbs up. That's great. So let's go ahead then. Seth, Seth sent me the squinty eyes. We shall see. Indeed, we shall. In not too much time, we'll see how we feel about those layers. So when we look at the skin, remember that we start with that keratinized stratified squamous on the outside. Then we go into the areolar. Then we go into the dense irregular, and finally finish off with that adipose tissue. All 4 of those layers inside the skin. So when we look at the skin big picture, we talked about in lab, and this looks just like our lab models, right? How we have the epidermis on the outside, the papillary layer of the dermis very small compared to that reticular layer. The reticular layer is where you have all those fun things, the accessory structures that make your skin work. Again, for my lab exam friends, let's do a little bit of review today. So I'm looking at a type of gland that dumps its secretions right onto a hair. What is the name of the special kind of gland we see here that's dumping its secretions onto a hair? Yeah, several of us typing in. It's a hard one to spell, right? These that I see on my hairs, and I see them over here as well, these are sebaceous glands. Sebaceous glands make sebum. Sebum, the stuff that we have too much of, right? When we have acne, we've got a buildup of sebum, this oily stuff underneath our skin. So sebaceous glands attach to the hairs. I have some glands that dump their secretions right onto the surface of the skin. What's the name of the kind of gland that dumps its watery secretions onto the surface of the skin? What were these ones called? Yeah, several of us chiming in. These ones are the eccrine glands. So the way you'll tell the difference is where they're found. Eccrine glands go to the surface of the skin. Sebaceous glands dump onto our hairs. We've got these fun little things right here, these little red things. These are called the erector pili muscle. What do these guys do? These red muscles here. I won't make you type their name. Erector pili. Exactly. Yep, these are the goosebump muscle. So they pull on the hairs, pull those hairs forward, which bunches up your skin, gives you a goosebump. We can also see in our picture here a couple of nervous tissue structures. So remember that down deeper in the skin, we have those things called the lamellated or the lamellar corpuscles. These are our pressure detectors. Up higher, we have those free nerve endings. This particular image, I don't think has what we had in lab called the Meissner's corpuscles. That was that gray thing that was at the top in our lab pictures. Remember that the Meissner's corpuscles were what helped us to do soft touch. So reading Braille is the best example of that. These free nerve endings that we see up here, these are things like itch and tickling, all those great sensations, right? So this kind of stuff. Oh, I apologize. Yes, Liza. So sorry. I was jumping into old school anatomy teacher mode. Meissner's corpuscles are called tactile corpuscles. We like to use the easy word. Yes. So sorry. In our lab, if there is an easier name for it, we will take that. So tactile corpuscle is correct. Ignore what Dr. Alice was saying about the other name of it. That's what I learned when I was back in the day in school with dinosaurs. But yeah, we're calling it the tactile corpuscles. So sorry. What I want to start with today, and where we're going to spend most of our time today, is in the epidermis layer of the skin, the outer layer of the skin. So I'm going to go through and talk with you about what makes the epidermis so protective for you. Remember from lab when we were talking about the different layers of burns, we said that one of the big problems with a 3rd degree burn is that literally there's nothing to block your inside from the outside environment. So the big job of the epidermis is to protect you from the environment around you. So we're going to spend some time now to chat about how it does that. The first way that your skin protects you from the environment around you is because of the keratinized stratified squamous epithelium. So that tissue that's on the very outside, in its name, it says it's keratinized, which tells me that these cells are full of keratin. One of the first ways that we protect ourselves from the outside is by filling our cells on the outside with this protein keratin. When we talk about the keratin protein, this is a really strong protein. It's a really tough protein. You have a bunch of it in things like your fingernails and your toenails, so it makes it very strong. When we look at the keratin protein and we see up close that protein, we're looking at our amino acid chain over here on the left, we can actually see part of what makes it so strong. Hey, so some protein structure review for us here. When I look at my keratin protein right here, I can see one of my kinds of secondary structure. What was the name of the secondary structure where we're in this curly Q ribbon? What secondary structure does keratin have? Ooh, maybe we forgot what our secondary structures are. Yeah, it was mean to make you type that too, right? So when we talk about our secondary structures, the 2 options are alpha helices or beta sheets. When I look at the curly Q ribbon, that's the alpha helix that goes around in a circle. When we talk about beta sheets, that would go back and forth kind of on itself, maybe fold a little bit like an accordion. Keratin has a bunch of alpha helices. That's its secondary structure. And that by itself isn't such a big deal, but what is a big deal about this secondary structure is this right here. I want you to underline, highlight, star the word hydrophobic. When we are looking at keratin, this protein with its alpha helices, there's a whole stripe or a whole row of amino acids when we fold it up into this pattern that are all hydrophobic. If I'm hydrophobic, how do I feel about water again? What do hydrophobic things think about water? Yeah, they don't play nice with it. They hate it. They loathe it. I love it. Yep. So one of the movies that Dr. Allis likes to watch every year at Christmas is Jim Carrey's How the Grinch Stole Christmas. I don't know if there's any fans out there, but he has this part in the movie where he's like, loathe entirely. So that's what I think of when I think of hydrophobic amino acids. They loathe water entirely. This matters for me. Oops, hearing a mic. Can we check to make sure we're all muted? Let me check as well. This matters for us because when we start building our tertiary structure, when we start building the 3D shape, we end up with this hydrophobic line, this red line that I see here, that goes all the way around this protein. And this line of amino acids loathes water entirely, so it finds a friend and it curls up with that friend. When we build a finished tertiary structure on a keratin protein, it's wrapped around each other kind of like a Twizzler. And I've got these 2 strings of amino acids that are really strong and stable because they both don't like water. So part of what makes the keratin protein so strong is it has this stripe of amino acids. They all want to push away water, so they squeeze together to make this strong protein. So first job of keratin protein, it fills up these cells and makes them really tough. It makes them resistant to damage. The other job of the keratin protein is to help us make a kind of attachment site between our cells called desmosomes. Desmosomes are a type of attachment site that resist pulling in multiple directions. When we talk about how keratin is involved in this, when we look at these types of junctions, see the little purple lines that I see here. These little purple lines are all keratin proteins. If you ever walk in to Walmart, for example, and you look up at the ceiling, how they have those rafters that go up to the ceiling in a line, that's the way that the keratin proteins work inside these keratinocytes. So if my neighbor cell starts pulling this direction, say I'm writing notes and it pulls my cell one way, the keratin proteins down here are like, oh, I got you. You're anchored to me. We're good. If this cell starts pulling this way, the keratin proteins over here are like, oh, I got you. You're anchored. No big deal. We put these keratin proteins in all different directions to anchor these two cells together so that they don't fall apart. If you remember, there was a layer of the epidermis, one of my strata, that we said looked like it had little pokey things on it, or it looked like it had little spines on it. Do we remember which layer of the epidermis I told you looked like it had spines? Which stratum? I'll do a really terrible sketch of the epidermis over here too. Which layer is spiny? Exactly. Yep. The layer that I am talking about is a layer called stratum spinosum. So I'm drawing you some really sad looking skin over here. So stratum spinosum, that's this layer right here. Stratum spinosum. Stratum spinosum, when we look at these cells on a slide, you can actually see the desmosomes that are attaching them to each other. So the reason this layer has the name stratum spinosum is because you can see these proteins that are holding together the cells using this type of junction called the desmosomes. So desmosomes, a kind of junction that we see a bunch in stratum spinosum. Stratum spinosum, the cells are still alive. The cells that I have in stratum spinosum, they started here. What's the name of that lowest stratum in the epidermis? What's the one at the very bottom? Exactly. Yep, the one at the bottom, the one in the basement, stratum basal. That's where all my cells come from. Stratum basal is the layer, remember I told you that we do mitosis, that my cells divide. So stratum basal, bottom layer. On top of that, stratum spinosum, where we have these spines, these desmosomes. Then remember that we have that layer that's dark above it. This one was called stratum granulosum. And we're going to talk much more about this layer here in a moment. If I'm doing the type of tissue that goes straight to stratum corneum, if I just have 4 layers, am I looking at thin skin or thick skin? If there's only 4 layers to the epidermis. Exactly. Yep. If I only see 4, so we've got basal, spinosum, granulosum, corneum. If that's all I've got thin skin. Thin skin is found on most of the places of your body. So most of your body is covered with these 4 layers. What was the name of that other special layer, though, that I have in my thick skin? What layer do I have there? I'm making my corneum look a little bit more cornified, a little bit more dead. Exactly. Yep, stratum lucidum. If I have that clear layer, stratum lucidum, that's one of the ways that I know that I'm in thick skin. In thick skin, we also see a really, really big stratum corneum. So stratum corneum would be really large, and we'd also see that clear layer of stratum lucidum. So a little tangent, again, from my Monday lab friends, because I know that we're going to have to know the regions of the skin, those strata of the skin. Back to our previously scheduled content right here with desmosomes. Desmosomes, tough junctions that resist pulling up and down, apart, all directions. Desmosomes are created with those keratin proteins, those proteins that make our cells really strong. And we can see desmosomes most clearly in that layer called stratum spinosum. Hey, can someone tell me in the notes, which page do we start talking about tight junctions? What page number is the tight junctions in our notes? I'm seeing page 3 and page 4. Okay, well, wherever it is that it first starts talking about tight junctions, I need everybody, and I mean everybody, to write this down for me, somewhere next to the discussion of tight junctions. Here's what we need to write down. No keratin here. No keratin here. Tight junctions are not made with keratin. They're made with a different protein that I'm not going to make you learn, but I need you to know for sure that I don't use keratin to build tight junctions. I use keratin to build desmosomes. It helps me to anchor my cells together in all kinds of directions. But when I talk about tight junctions, I do not use keratin for that. Tight junctions are my connection point that helps me to move together two plasma membranes so close that they're basically fused. So the goal of a tight junction is to waterproof your skin, to make sure that if there's some water out here next to my cells, that water can't just sneak down in between my cells and go into my body. Tight junctions anchor together the membranes. They make it so there's no space for water to move in and out. Again, I can't emphasize this enough because I'm promising you it's going to show up on the exam. I do not use keratin to build tight junctions. I do use keratin when I'm building desmosomes, but not these guys. So here's what we've talked about so far. Thing number 1, keratin has that twisty pattern where we end up with the licorice. Keratin is a really strong protein. I fill the cells on the outside of my skin with this protein to make them strong. Number 2, keratin is what I use to build desmosomes, to build attachment sites that I can pull in any direction and they'll stay attached. Number 3, we talk about tight junctions. These are my waterproofers. These make it so there's not space for water to sneak in between my cells. But the tight junctions are not the only way that our skin is waterproof. The other thing I use to help me waterproof my skin is something called glycolipids. Glycolipids are sometimes called lamellar granules. I'm going to type that in the chat for you. Lamellar granules. Lamellar granules, or glycolipids, are the reason that stratum granulosum has those purple spots. Those purple spots that you see in stratum granulosum are these things called glycolipids. So when we talk about glycolipids, their name tells us some stuff about it. They've got the glyco part, the sugar part at the top, and then the lipid parts down at the bottom, the lipid tails. The chemistry of glycolipids is mostly determined by these tails right here. So remind me in the chat, when I talk about things that are lipids, they do or don't like water. Lipids do or don't like water. Correct. Lipids, whether they're the phospholipids in the membrane or whether they're the glycolipids that we see here. Lipids do not play nice with water, which is why we use glycolipids to waterproof the skin. So here's how it goes. I'll grab my pointer. I'm a cell that lives down here in stratum basale. I'm happy, I'm dividing, I'm pushing up into stratum spinosum. I'm growing, I'm building keratin in stratum spinosum. Eventually I end up here in stratum granulosum. By the time I get to stratum granulosum, I'm realizing, oh, I am getting close to the outside of the skin. I kind of go through a midlife crisis and say, what can I do to protect myself when I go to the outside of the skin? And then I have this revelation, I could make these things called glycolipids. Glycolipids would keep the water that's outside from coming inside. They'll keep me safe. So in stratum granulosum, my cells start to make these glycolipids. You can see them, these little purple spots are all glycolipids, and they spit them out outside of them, and it puts a raincoat on them. They're blocked from the outside. That's good news when we're talking about water coming in from the outside. That's bad news when we're talking about the water that's coming from the inside trying to go up. When I talk about the water on the inside trying to go up, what kind of things do you think that the water, aka the blood, would have in it that these cells want? What does blood have that your cells need to survive? Exactly. Yep, things like oxygen, things like my favorite foods, glucose. When I put on a raincoat and I block any water from the outside, I also block water from the inside. And water, or plasma, is how I transport glucose, it's how I transport oxygen, it's how I get things around in the body. So stratum granulosum is the layer when the skin cells start to die. They're dying because they're literally starving to death. Below here, we're alive, we're getting the nutrients from our blood supply. Above here, this layer and above, these cells are dead because we've got a waterproof barrier that's preventing the oxygen and nutrients from getting up. Hey, by the way, I forgot to share this with you in lab. So here's a little nugget to help out with your lab exam. When we're trying to remember the order that the layers go in, hopefully you're good on that. But in case you aren't, a mnemonic that I have seen that was really good was, come, let's get sunburned. Oops, sunburned. Stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and stratum basale. Come, let's get sunburned. Or if we're talking about thin skin, like we see in our picture here, it's just come get sunburned. I feel like that should be the sign next to NRH2O, right? Come get sunburned. So to help you out when you're remembering those layers, think of that statement going from the outside to the inside. Remember, anything at this level or above, stratum granulosum, all of this stuff is dead. And it's dead because of the glycolipids. The glycolipids block us from sending things up, from sending nutrients and oxygen above. I want to pause for a moment to give you a chance to ask questions or to give me a thumbs up. How are we doing with the epidermis? Either we all fell asleep or Dr. Ollis has some serious lag. Hannah's thumbs up. Perfect. Okay, well, if you think of any other questions, always feel free to slide them into the chat for me. I'm going to keep us going because I have just a few more things I want to talk about during today's session. I want to talk about these cells that I see right here. This is another kind of cell that we find in your skin, though it's not one that we had you learn for lab. This is a kind of cell called a melanocyte. And the name melanocyte tells us what these cells make. Melanocytes make melanin. And melanocytes are the only kind of cell in your skin that have the ability to make melanin. Melanin helps to protect us from UV radiation. And UV radiation is bad news because it comes in and it changes the sequence of your DNA. Now, I'm hoping after our discussions last week about things like sickle cell anemia, you have a pretty good idea of why it's so bad if your DNA sequence changes. So the goal of melanin is to prevent your DNA sequences from changing in your skin. It makes an umbrella to block the UV radiation. When we look around, granted we're not in a classroom together, but if we were in a classroom together and we were to look around our classroom, we would see quite the variety of different skin tones. When we talk about having darker skin, having lighter skin, what I need all of us to write down is that we all have the same number of melanocytes. All of us have the same number of melanocytes. Whether you're over here on the fair end of the spectrum, basically translucent skin, like Dr. Allis, or you're over here on the darker end of the spectrum, we all have the same number of melanin-producing cells, but we don't all have the same amount of melanin. Some of our melanocytes are really good at protecting us from UV radiation. They make a lot of melanin. Some of our melanocytes, like mine, are a little bit lazy, and they don't hardly make any melanin for us. We all have the same number of melanocytes. Some of our melanocytes are more active than others, which means that we'll have more melanin in our skin. It is possible that you can actually increase the amount of melanin that your skin is making. We can increase the amount of melanin that our skin is making by purposefully exposing ourselves to UV radiation. The normal person word for what we're doing when we purposefully expose ourselves to UV radiation is tanning. So when we go to a tanning bed, when we lay out next to the pool, when we're chilling out at the beach without our sunscreen, what we're doing on a physiology level is we're exposing our skin to UV radiation. When we expose our skin to UV radiation, the melanocytes say, oh, we are not safe right now. Let's make some melanin. Let's spit it out to cover everybody up, to keep everybody safe. Keep in mind, though, melanocytes can work somewhat quickly. They can build melanin relatively quickly, but you still always started with an initial UV exposure. We had the potential to damage our DNA to get those melanocytes to get active. So Dr. Allis is a little bit on a soapbox here with tanning and UV radiation because for about 1.5 years, I worked for a skin cancer dermatologist. So we spent 3 days a week. All we did was skin cancer surgeries. So Dr. Allis says, I know that being a bronzed goddess or a bronzed god is desirable, but please don't sacrifice your DNA to get a little bit tan. Again, big idea with this. We all have the same potential to make melanin. We all have the same number of melanin-producing cells. How much melanin we produce, that comes down to gene expression, like we talked about last time, when our RNA polymerase attaches and makes that gene. expresses the melanin gene. We have the potential to do it the same, but we don't all make the same amount. Yeah, so Danae is agreeing with me as an esthetician. Yep, wear your sunscreen so that you don't have to go see Danae and she has to do some serious work on you, right? To UV radiation, man, it doesn't just give you skin cancer. That's like worst case scenario. It also is really tough on your skin. So Dr. Ellis will be the first to admit that I need to, when it comes to summertime, I need to, well, should be doing it all the time. I need to be wearing my daily sunscreen to make sure that I'm safe. But trust me, when I go to NRH2O or I go outside for any extended period of time, the sunscreen is on. My melanocytes are way too lazy for me to do it any other way. All right. Last thing I think I want to chat with you about today. Oh, here we go. I got to toss this up. I can't remember if I put this in the guided lesson or not for you. So here's my tanning cartoon. Keratinocyte over here. What's wrong, melanocyte? Melanocyte says she's tanning again, and I'm crying little melanin tears. So when you go tanning, you will make more melanin, you will get more tan, but you're also making your melanocytes cry. So don't make melanocytes cry. All right, let's talk about problems with melanocytes. So I think this is probably one of the last things we'll chat about today. So first problem that we can have with melanocytes is a condition known as albinism. So let me type it in the chat for us here. Albinism. Individuals with albinism have melanocytes, but their gene for the melanin protein has been mutated. So if they try to build the melanin protein, they cannot. So the melanin protein, we can tell from these pictures, it's a major player in skin color, absolutely. But the melanin protein also dictates things like hair color or eye color. So these individuals don't have the ability to make melanin anywhere in their body. Everything that normally would have melanin does not. These pictures that I included here, by the way, these are not people in makeup. These are not Photoshop. These are actual individuals with albinism. Albinism can affect anybody, any race. It just comes down to, did the gene for melanin get mutated? For my individuals with albinism, we can't make the melanin protein. What do you think that makes us much more likely to have problems with? If I can't make any melanin, why is that risky? Yeah, so Amush mentioned in the chat correctly, remember that melanin is supposed to be your umbrella. It's supposed to be what protects us from the sun's UV radiation. So absolutely, like Dusty mentioned, you get sunburned really easily. And even more than just getting sunburned, individuals with albinism are at a much greater risk of developing skin cancer. Exactly, like Brianda mentioned. Albinism, individuals with albinism, depending on how severe their albinism is, many of them cannot spend hardly any time at all outside because they have no protection at all from UV radiation. So really good sunscreen, protective clothing all the time. They have to be careful as well of UV exposure into their eyes because they have no protective melanin in their irises. Melanin actually also, we'll see this in lab at the end of the semester, there's melanin inside your eyeballs. So a lack of melanin, really bad news. The route we're going to go with it is it highly predisposes you to skin cancer. So when we talk about skin cancer, there's 3 types. The different types of skin cancer are named based on where they started in the skin. So let's start with the type of skin cancer that develops in stratum basal. So remember that stratum basal is the bottom one. It's the basement one. If one of my cells that lives in stratum basal is exposed to UV radiation, its DNA gets damaged, and then it starts to replicate way too quickly. That's what happens in cancer. We just keep making more and more and more of these cells. If I make more and more of these stratum basal cells, I have what's called basal cell carcinoma. Basal cell carcinoma. Basal cell carcinoma is the most common kind of skin cancer, but it's also the least deadly. Basal cell carcinoma grows relatively slowly. And when it grows, notice how at the bottom here, I kind of have a uniform boundary. So I told you I worked for that skin cancer dermatologist. We would do a surgery called Mohs surgery. The goal was to take out as little skin as possible. So the doctor would start, she'd go in and she'd make a cut, and she'd send that tissue to her histologist and say, hey, did we get all the cancer cells? And the histologist says, no, I still see some cancer cells down here at the bottom. So she'll go in and take another cut of skin. Did I get them all? No. Another cut of skin. Did I get them all? Oh, yes, we did get them all. So we were able to just go straight down in this area. It's very easy. It's called clear margins, where we catch all the cancer cells. Basal cell carcinoma grows slowly and it grows uniformly. All the cells grow down at the same rate. It's easier for us to take it out. It's harder for us to take out squamous cell carcinoma. Squamous cell carcinoma, the next most common kind of skin cancer, squamous cell carcinoma develops when one of my cells in stratum spinosum, one of my squamous cells, becomes cancerous. When these cells become cancerous and they start to divide, they don't divide uniformly. Notice how we see over here, we've got this little tendril of cells here, we've got some cells that are over here. Squamous cell carcinoma doesn't grow in that uniform pattern, and it grows faster than basal cell carcinoma does. So squamous cell, a little bit more serious than basal cell carcinoma. The most serious skin cancer of all, though, is melanoma. And when we talk about melanoma, this is the kind of skin cancer that develops when one of my melanocytes, one of my melanin producing cells, gets its DNA damaged. And see, that's the scary thing about tanning. Melanocytes build melanin for all the cells in your epidermis, but they're kind of like those people, maybe you're one of them, that just keeps giving and giving and takes care of everyone else, but doesn't really take care of itself. That's kind of the way melanocytes are. They'll give away the melanin to everybody to make sure everybody else is covered, but their own DNA is susceptible to damage as well. So if a melanocyte gets damaged, if its cells become cancerous, we develop melanoma. Melanoma is the fastest growing, and you can see in this picture here that it grows irregularly. The other thing about melanoma too is that it likes to spread. It likes to go to other parts of the body. So we'll talk more about spreading on Wednesday. The word for spreading is called metastasis. Metastasis. Melanoma is really the only skin cancer that typically metastasizes, that goes to other places in the body. And it can be very dangerous when it goes to other places in the body. If you are interested in learning more about melanoma, there are definitely some articles in Current Events and Anatomy that specifically relate to skin cancer, that relate to melanoma. So if you're more interested in learning about this, consider for Current Events and Anatomy picking up one of those articles. I'm going to save my stuff about staging melanoma to next time. Here's what I want to end with, though, to help out my lab exam friends today. So I'm looking at a sketch. It's not a tissue slide, but it does cover the same kind of stuff that our models and that our tissue slides do cover. So we can use that mnemonic, that phrase that we talked about before. I'm going to draw in my little levels here. All right, so I am looking at skin here. First question, skin that has 5 strata in it. Is this skin thin skin, thick skin? If it has 5 strata, 5 layers. Yes, I am looking at thick skin. What was the name of the specific layer that is only in thick skin? Which of these is only in thick skin? I've got their names over here. Who's only in thick skin? Exactly. Yep, this one right here, stratum lucidum, only in thick skin. Up above stratum lucidum, we have that layer called stratum corneum. Remember, the cells in stratum corneum are completely filled with keratin, and they're completely dead. They're falling off. Right here, I see those glycolipids. I see those little spots inside my cells. Can you remind me again, what's the function of glycolipids? What do those little spots do for my skin? Why are they helpful? Exactly right, Dusty. Yep, my glycolipids are my waterproofers. So stratum granulosum, where I have those glycolipids, those waterproofers. Remember that down below it, we've got that layer where we would see all those desmosomes, those connections that look like spines. Stratum spinosum. Remember that stratum spinosum, the cells are still alive. Stratum granulosum, they start to die. And then down here at the very bottom, stratum basal. Notice that stratum basal is the place where my melanocytes live. This funky looking cell with all of its little arms that are reaching out, all of those little arms allow my melanocytes to share their melanin with other cells. So the melanocytes live in stratum basal, but they share their melanin with cells everywhere. Stratum basal with keratinocytes and melanocytes. That's as far as we're going to go today. Are there any questions that we have before we call it a wrap? Or shoot me an emoji or a GIF. What do we think of the skin? Let's see what I can find while we're typing. Oh goodness, I have to send this in. I used to talk a lot about this lady here. I just sent you all a GIF of her. The lady I just sent in is Tanning Mom. Tanning Mom got in big trouble for taking her, I think she was 5, her 5-year-old daughter with her to go tanning. So she got in big trouble for that. I have not had a lot of time lately to talk about her, but yeah, Tanning Mom shows us what happens when we expose our cells to UV radiation. We do get tan, but if you notice, her skin also looks really, really aged. Tanning is bad on your skin for aging. So among other reasons, please don't go tanning, and definitely do not take your children with you when you go. This public service announcement brought to you by Dr. Allis. All right, well, I'm going to go ahead and stop the recording. I will stick around in the chat for any last minute questions that you have. Otherwise, if you are one of my Monday lab friends, I'm going to plan to open up the lab room at about 9:25. So if you're here now, you can head over this way. But yeah, by 9:30, make sure you're in your seat for us.